Today we introduce a paper from Nature titled, "The Microbiota Regulate Neuronal Function and Fear Extinction Learning." Pavlovian fear conditioning is an evolutionarily highly conserved cognitive process that enables organisms to assess whether external stimuli are safe or dangerous, significantly enhancing survival. Fear extinction learning refers to the situation where a stimulus signal appears but is not accompanied by any danger, so the body does not respond with avoidance behavior. Abnormalities in fear memory extinction are associated with multiple neuropsychiatric disorders. Furthermore, numerous clinical and epidemiological studies have linked changes in the microbiota to various neuropsychiatric conditions. Consequently, the authors hypothesize that abnormalities in fear memory extinction may also be interrelated with the microbiota. The following research confirms that fear extinction learning in mice requires the participation of the microbiota. First, the research team conducted behavioral experiments using three distinct mouse categories: normal mice, adult mice treated with antibiotics termed ABX mice, and adult germ-free GF mice. These experiments demonstrated that signals from the microbiota are essential for optimal extinction of conditioned fear responses. Subsequently, vagus nerve resection demonstrated that extinction learning deficits in ABX mice are independent of the vagus nerve. Analysis of immune cell composition further revealed that the adaptive immune system is not required for extinction learning deficits in ABX and GF mice. The team then isolated MPSC cells and performed RNA sequencing, identifying substantial transcriptional differences between the two groups. These differentially expressed genes clustered around neuronal activity, synapse function, CNS maturation, and synaptic plasticity. To define the cell subsets in the MPSC that contribute to the effect of the microbiota on extinction learning, the research team performed single-nucleus RNA sequencing SNRNA-seq of MPSC samples dissected from ABX and control mice after extinction learning. Results showed that excitatory neurons exhibited more pronounced changes than inhibitory neurons. The most pronounced changes occurred in three types of supporting cells: astrocytes, myelinating oligodendrocytes. Finally, the research team investigated the effects of microbiota at the histological level. They noted that postsynaptic dendritic spines undergo dynamic changes during learning and memory processes. Using two-photon laser scanning microscopy, the authors discovered that compared to normal mice, ABX mice exhibited a significantly increased rate of spine loss, while the formation rate remained largely unchanged. Furthermore, while no significant differences were observed between the two mouse groups in the fear conditioning model, distinct differences emerged in the fear memory extinction model. Normal mice exhibited increased dendritic spine formation rates, whereas ABX mice showed minimal change. While normal mice demonstrated little change in spine loss rates, ABX mice exhibited a sustained increase. These results indicate that the abnormal fear memory extinction induced by the microbiota is associated with alterations in synaptic dendritic spines. In summary, this study compared behavioral differences between normal mice and antibiotic-treated mice, revealing that mice with cleared microbiota exhibit abnormal fear memory extinction. Subsequently, differences between the two mouse groups were investigated at molecular, cellular, and tissue levels, thereby establishing the mechanism by which microbiota influence fear memory extinction in mice. That's all.